welcome everyone back to another session of this webinar series on scientific machine learning. I'm excited to continue this five-part series on molecular machine learning for drug discovery. I'm particularly pumped up about today's session because we have Bharat Ramsundar from Deep Forest Sciences presenting today, who I closely work with. And for the first time, we have a rising star, 18-year-old researcher, Seyon Chitrananda from the University of Toronto. We also have Gabe Grand from Revere Labs. So our session chair will introduce Bharat and Seyon shortly, but before that, let me introduce our session chair for the day, Tom Miller, who is a scientist and entrepreneur with focus on AI-driven methods for molecular simulation and discovery. Following a PhD at Oxford and a postdoc at UC Berkeley, he joined the Caltech faculty in 2008 and has been a full professor since 2013. He has received awards for research excellence from the Sloan Foundation, the Dreyfus Foundation, and the American Chemical Society. He has mentored dozens of students and postdocs that are now leaders in academia and industry. In 2020, Tom co-founded Entos Inc. to accelerate molecular and materials discovery via physics-based machine learning. Tom, welcome to the session as the chair, and thank you very much. With that, over to you to introduce Bharat, Seyon, and Gabe.
Wonderful. Thank you so much, Dilip, and thank you also, Venkat, for the chance to participate in this exciting series. Today looks like a fantastic seminar and discussion, and a chance to learn from two real leaders in the field. Bart Ramsundar is the founder of Deep Forest Sciences, which is an upcoming research and development company that leverages his Deep Chem package to build AI for deep tech problems. Bart did his PhD in computer science at Stanford University in the lab of Vijay Pandey, where he studied the application of deep learning to problems in drug discovery. At Stanford, Bart created the Deep Chem IO package and co-created MoleculeNet.ai to grow the Deep Chem discovery open source community. Bart's also a Hertz Fellow, the most selective graduate fellowship in the sciences. Seyon is an 18-year-old working in the intersection of machine learning and biochemistry. His research focuses on cutting-edge applications for drug discovery using machine learning as a research intern in the Matter lab at the University of Toronto, supervised by Professor Alan Asper-Guzik. He's also an open source developer for Deep Chem. His work is supported by the Emergent Ventures Fellowship from Dr. Tyler Cowen and the Thiel Foundation. So today we're going to hear a presentation from these exciting leaders in the field, the title of which is before us, and we'll focus on the ChemBERTa infrastructure for machine learning and molecular property prediction. So Bart, Seyon, over to you.
Okay, awesome. Thank you for kind of the kind intro, Tom. I'll also want to give a shout out to our co-author, Gabe, who will be helping us on kind of during this presentation. Thank you all for dialing in today and listening. We'll primarily be talking to you about our new architecture, ChemBERTa. And before we even dive into the presentation, just to give a brief, almost a TLDR for the presentation, our goal really is to explore the use of natural language processing based techniques for molecular property prediction. And primarily, we want to see whether we can adapt pre-training techniques, which in NLP, if you've ever had the chance to play with some of the new OpenAI tools, you know just how powerful these systems can be. By reading the entire internet, it's almost achieved the intelligence of a Reddit poster, which, it's I won't say it's a very high bar, but it's definitely a bar that has been reached. So can we achieve the Reddit poster level of chemist? That's our goal today. Just set the stage for what we're trying to do. But joking aside, we think there's really cool techniques that can be brought in from the NLP community to apply to chemical applications. We won't say too much in this presentation about it, but similar techniques have also been applied to a protein language model. So there's, I think, a broad spectrum of techniques from natural language processing that are beginning to find their way into the sciences. And we hope that we can provide a bit of an introduction to these and other techniques, and also just discuss this new architecture and this new way of doing molecular property prediction that we're very excited about. Okay, with that, let's go ahead and dive right into it. Next slide, please. Yeah. Okay, so for those of you who worked in kind of drug discovery, you've seen this slide before. For those of you who haven't, Eroom's law is essentially the opposite of Moore's law. Like, it's really Moore's law spelled backwards. You know, for decades, computer chips have gotten cheaper and denser year over year, and just in parallel, for decades, finding new medicine has gotten more and more expensive and more painful. Now, there's a variety of reasons that this could be happening. One could be, needed regulatory burden. There's kind of a famous incident with thalidomide, which unfortunately caused birth defects in many children, which led the American FDA to really start tightening up regulations. And globally now, with kind of all the other regulators, there is a rising bar just from as we learn more about safety, as we kind of learn more about the potential long-term effects of drugs. There's some needed tightening there. But at the same time, it's also possible that perhaps we plucked a lot of the low-hanging fruit. Maybe a lot of the obvious things have been found, and we need new ideas, kind of new techniques to really break the gridlock in drug discovery. Stepping back a bit, we've all seen this year in the pandemic, like one of the few bright spots has been, say, Moderna, Pfizer, BioNTech, just really rushing a vaccine through in, amazingly short period of time. Like, what is it, 10 months? So we know now that we can actually do faster when, the fate of the world is at stake. And a question I have for all of us, and I know for like the other speakers in the series, I know some of the work our chair is doing, like how can we use new techniques from software to speed this up, to make it possible to find medicine more quickly and safely, and make a difference in the everyday. Next slide, please. So, finding a new medicine is a long and complicated process. Here's just like the very rough skeleton pipeline. Let me just try to step back and give, the 10,000-foot overview of the process of finding a new disease. As with all good science, we often start from a cartoon model. We try to put down some type of reductionist model of what causes the disease. We say something like, well, the real disease in humans is complicated, but I'm going to form a reductionist hypothesis that the primary driver is, say, an issue in this one protein. And to clarify, for real-world diseases, there are often multiple potential targets, potential proteins or protein-protein interactions that we could modulate. But it's useful to think about this as, let's just make like a very simple cartoon diagram. We say, if I could turn that off, if I could modulate it, then all of a sudden I will cure this disease. That's a hypothesis, the lie we tell ourselves to start. Then we say, okay, I've decided on this convenient, beautiful illusion. Let me now try to act upon it. Can I find a first compound that modulates that protein in question? This is kind of the hit finding. A hit is that first, compound which can have some activity. Then you say that, well, to use a crude example, if I poured bleach on a cancer cell line, I would, of course, destroy the cancer cell line, but I would do nothing good for a human patient. Let's turn this molecule into something that is closer to being safe for humans. So one way to do that is to make sure that the molecule interacts tightly with, say, the protein in question. You might also want to model things like toxicity, metabolism. This is called ADMET, absorption, distribution, metabolism, excretion, toxicity. We want to the best of our knowledge, know that the potential compound we're thinking about is safe for humans. We will then kind of move to animal trials. This is, I'll step back. This is nothing to do with our talk, but one of the hardest problems, I think, in modeling disease is that humans get diseases that animals don't. This is particularly difficult in things like neurological conditions where mice are not as smart as people. This is why we don't really have an Alzheimer's or Parkinson's drug that really works yet. But stepping aside, again, convenient, beautiful illusions. Let's pretend, or let's make do with the best animal model we have for the disease in question. Let's give it to the animal. Sometimes it works, sometimes it doesn't. There are some very good drugs that have failed at the animal models because, say, humans are different from rats or cats, but they've been rescued. But let's assume that the model works. And then finally, we say that, okay, we need to scale this out so we can actually begin running larger clinical trials where we're going to give this to real patients. So this is a really useful skeleton to keep in the back of your head. All the work that we're doing, that other people in this field are doing, are really, I think, guided by this picture in the back of our heads that we want to find ways to speed up various parts of this process. And the work that we will present on in this talk in particular, I'd say is the middle three bullet points, covers the middle three bullet points. Can we use these new techniques of natural language processing driven molecular property prediction to speed up hit finding, to speed up lead optimization and ADMET modeling? But with that, let's kind of dive back into some more things. Next slide, please. So another kind of point about the industry that's worth pointing out, pardon me, it's early here in the morning, so I have not had my morning caffeine really dial into me there. Proprietary techniques are standard. A lot of the biotech industry is driven by IP, intellectual properties like the lifeblood of a company. If you've ever had the fortune or misfortune of wrestling with a biotech company lawyer, you will know that this is something that people care intensely about. And for good reason, but this does sometimes make it challenging to do science. Patents cover a lot of the core techniques in the space. Things like CRISPR, of course, have had long-running patent battles as people try to figure out who owns what and who gets the royalties. This has also transferred somewhat to software vendors. Traditionally, software vendors have maintained pretty tight control over their software. Think things like per-user site licenses. This can make it challenging for students or hobbyists, or say new researchers who are outside, say, the core Boston, Bay Area, UCSD, et cetera, axes of drug discovery. And this is, I'd say, in marked contrast with the way the rest of the tech industry is heading. Next slide, please. So we have begun to see what I would say is a shift towards software-driven discovery, which is driven by the advent of a variety of new techniques, a lot of them driven from the molecular machine learning community, a lot of them driven from other aspects of, say, like software more broadly, like just the fact that our tooling for a lot of modern software is dramatically better than it used to be. Sorry, next slide. So some of the techniques that people have started using are to use, of course, molecular machine learning to speed up the algorithmic design of molecules, to be able to hopefully take the steps that we talked about previously, things like hit finding, things like ADMET modeling, and be able to run that more effectively and more quickly directly in the in silico, as it were, rather than in a lab. I will also say that a lot of these techniques are still in very early days. They're just now beginning to shift out of the research lab and to practitioners. Next slide, please. And another kind of trend I will mention here briefly is that, of course, if you worked in kind of modern tech, you'll know that a lot of it is increasingly driven by open source software. So you have powerful frameworks, things like React, TensorFlow, PyTorch, Postgres, and all of these are run by these open source communities that I think really form a network that connects a lot of the modern tech industry. You might say go from Google to Salesforce to maybe even Genentech, and you might end up, say, using TensorFlow throughout that process. It creates a common layer of shared utilities and shared know-how that I think has helped bootstrap a lot of the tech industry. But the challenge here, of course, is that this contrasts with what we just saw with how the pharmaceutical industry traditionally works. And next slide, please. And this, I'd say, is really the impetus for behind the Deep Chem project. Can we somehow leverage the superpowers that systems like Linux have given to the modern tech industry and bring that to the biotech world, bring that to the process of medicine discovery, make it easier for people to share techniques and know-how across projects and across companies? Next slide, please. So what we've been trying to do with Deep Chem has covered a number of different axes. Of course, we want to establish open source software that makes high quality open tools for deep learning. We want to also, and I'll say more about this later, establish open source data sets. A lot of machine learning really is driven by access to clean, high quality, standardized data. There's limits to what data you can find in the open source, but the easier it is to get access to high quality data, the faster machine learning develops. So we also maintain and expand upon the MoleculeNet benchmark suite for molecular machine learning. And also we maintain a large collection of free educational material. So there's an extensive collection of, tutorials. There's also lots of documentation, even a book out there for learning to use Deep Chem and applying it to your projects. Later in this talk, Seon will present one of our new tutorials for this work, ChemBERTa, that'll allow you to actually run alongside us and try these techniques out for yourself in your browser through Colab. Next slide, please. So, if you're curious, you can check it out online. Go to deepchem.io. Next slide, please. And again, here's just a picture of the MoleculeNet benchmark suite that I mentioned earlier. This is a diagram from our original paper a few years back. The benchmark suite has actually grown, probably doubled in the years since. We're working on a sequel to expand this out even further and kind of bring this more members of the community to participate in maintaining a communal benchmark. All of this is accessible through Deep Chem. If you install Deep Chem, like pip install Deep Chem, you can actually just download a package from the MoleculeNet benchmark suite with kind of one line. You do something like deepchem.molnet. Pick a package, load Tox21, and bam, you're off to the races to be able to start benchmarking. Next slide, please. But okay, with that, there's a lot of background information, a lot of just things about the industry, things about drug discovery and software that we covered. But now let's actually get into the molecular machine learning. So I'd like to just set the stage a little bit for why we're working on this problem, why I think other members of the community are working on this problem. And what I'll say here is that we say that transfer learning is something of a grand challenge problem for molecular machine learning. And what I mean by that is that in a lot of machine learning, it's really easy to get data. If you have ImageNet, for example, it was actually strangers on the internet, coordinated through Mechanical Turk, who labeled pictures of cats and dogs, and were able to create the training data set for ImageNet with relatively cheap cost. Whereas there is no collection of strangers on the internet that can really annotate a experimental data set for chemical applications. You actually need a lab. You need trained technicians and scientists. And this challenge, I think, is true for not only for us in molecular machine learning, but true for scientific machine learning more broadly. We have and will struggle to handle data more effectively, more than other machine learning communities. So for us, I think transfer learning, which is roughly defined, can we find a way to transfer in information from other sources to apply to a problem that we have at hand today with limited data, becomes really important. Now, the challenge here is that just because the problem is important doesn't mean it's easy. In fact, transfer learning, I'd say, is terribly difficult. If you've tried using any system that purports to bring in other information and apply it to a new context, you'll know that these systems are brittle. They don't work. We've seen kind of some examples in machine learning where transfer learning has begun to work. If you look at, again, going back to GPT-3 or these OpenAI systems, if you look at, say, ImageNet or these image recognition systems, we do actually begin to see effective transfer learning. Now, for molecules, though, it's a bit of another story. Next slide, please. So to provide a little bit of just framing for why, put some numbers on kind of the brief introduction we mentioned there. So there's a dramatically increasing scale of the amount of data that you get as you kind of loosen the requirements on labels. When you're looking at high quality experimental data, you're often looking at hundreds, thousands of data points, especially if the assay in question is challenging to run. There are some larger robotic assays where you can get tens of thousands or even hundreds of thousands of data points, but it's often a limited biological readout. So essentially, the more high quality the data, the lesser amount of it you have. And there's an inverse relationship where as you loosen the data gathering requirements, you start to get more data, but it's not until you go to essentially unlabeled data that you start seeing abundant streams. So there's now, combinatorial chemistry data banks of molecules like, enamine, zinc 15, where these are molecules that are very likely to exist, but may not even have been synthesized, but have been algorithmically generated, that we can begin to use as something as, the background Reddit noise of the internet, so to speak, to train one of these models. Can we learn a prior on chemical space by using these large unsupervised data banks? And if we can do that, perhaps that's a way to bypass or at least loosen this grand challenge we face with transfer learning. There's a lot of these molecules out there, and if we could somehow impute enough chemistry within the model, then perhaps with even 10, 20 data points, we can start to do interesting things. Next slide, please. To be totally kind of upfront, there is a long and large literature of people who've tried this. We've tried variants of techniques earlier in Deep Chem. There's some notable work from a number of other groups. Recently, Yuri Leskovic's group at Stanford has explored graph-based pre-training. We'll come back, say a bit more about that shortly. There's a lot of techniques people have tried: multitask learning, one-shot learning, semi-supervised contrastive learning. I'm probably missing a whole host of literature. This is a hard problem. A lot of people have tried. None of these techniques really work yet. As a spoiler, our technique doesn't really work yet, but we have some reasons to at least find hope for optimism that these NLP techniques, I think, can help us take a closer step towards effective transfer learning in this space. Next slide, please. So to provide just a little bit more context, like this work and a lot of the other similar projects have been really inspired by the rise of these transformer-based BERT-style architectures for natural language processing. In the last several years, we've just seen a dramatic boost in NLP applications across the board, from language modeling, say reading comprehension, machine translation, that have been driven by these transformer architectures. If you haven't already, would recommend checking out a GPT-3 demo from OpenAI. I mentioned them a few times because I'm just very impressed by the quality of what they've done with very large scale training. And the hope is, can we bring this to molecular machine learning? And with that, next slide, please, Seon. I'm going to pass the mic over to my colleague, Seon, who will take us through the ChemBERTa architecture and will also guide us for the demo there. But before I do that handoff, Tom, are there any questions or things for us, since it's kind of a bit of a pause point?
I think it's a fine idea to pause and to take any questions from the audience if they would like to do so on the basis of this. And it's also a chance for me to make sure that I round out our welcomes from the initial part by giving a nod to Gabe as well, who's joined us. Gabe Gren, thank you also for joining us. He's interested in the multimodal learning problems at the intersection of computation and cognition, and has joined us. So if anybody has any questions, please raise your hand or speak up, and we'll be happy to receive those. I think it's been a very...
Can I ask something?
Yeah.
So Bharat, I think you made a very interesting point about ImageNet, sort of as a democratized way of generating large volumes of labeled data. Have you thought through how? And this is something I've also sort of thought about from the energy storage context, where what will it take to sort of build a BACnet or whatever. And how do you see this happening in sciences? Like, is there a way in which there's you could do sort of democratized generation of data?
That's a really, really good question. I know in chemical applications, there has been a rise of, I'd say, like superior chemistries that are capable of scaling up more. Notably, like DNA encoded libraries, I think, have started adding these very large data sets that are more accessible to kind of people to run at scale. I think the naive answer would be to say roboticization, but I don't actually think robots are necessarily that useful. I think instead, like better experimental techniques that enable scaled readouts, even with humans running the assays, might begin to really make a difference in the short run. But it's a really hard question because like this, of course, really depends on the field in question. Like the experiments needed for good battery data are probably, or maybe really different from the experiments needed for good, say like drug discovery readouts. So it might, but I do think that a large part of the community in these fields is already thinking about, students are kind of coming out now having taken multiple machine learning courses. They know that, hey, we need more data. And then naturally things start to rise where people think about the scale issue, which that's new. I think in the past, people were like, why bother like creating these data sets? It's like, let's focus on the science. But I think there's an increasing awareness that the creation of larger scale data gathering techniques is a science in its own right that will bootstrap a lot of these AI techniques. That was a bit of a non-answer, Venkat, so my apologies, but it's a hard question.
Can I follow up on that? I mean, you'll, of course, be aware of efforts for federated learning where different companies will take their proprietary data, anonymize it some way, and then pool it for the greater good. And it does seem like there's a lot to be gained through making people understand that by giving a little, you get a lot back. And do you see that that could be a continuing trend? Do you see ways in which that could dovetail with some of these efforts that you're focusing on the software side here?
That's a great question, Tom. Yeah, I do think it can definitely play a role, and I think it can be like a powerful complement. So I've been involved with a few efforts with like back in grad school, we trained a model on some of obfuscated fingerprints from Merck, where it was the only way they could send it over the legal firewall to us. There's at my last startup, we tried things with like experiments of data pooling and incentives for gathering data from different sources. What I will say is that I think it's a really hard problem. Like I'm interested and I'm continuing to hope that architectures for this come about. I know that there's also like increasing emphasis on privacy, and GDPR regulations have been driving, I think, a lot of innovation in the space. I've not yet seen like a chemistry application that's really blown me away, but I think that's perhaps just a call for like figuring it out. So I think it's a great idea. I think it has a lot of potential, but I haven't seen anyone, actually, same as with transferring, I haven't seen anyone really like knock it out of the ballpark yet. Yeah, I think it definitely has a role.
Great. Well, Rachel Kirchin has also submitted a question, which I'll read. In the material science space, a big issue that goes along with a much tighter cost of data collection is the issue of data standardization/curation. Loosely speaking, making sure that it's actually valid to compare numbers in one database to another when they have been collected or calculated in different ways, and including tags of some sort if we can't compare directly. Her question is that I assume there are lots of analogs to this in the drug discovery space, but I'm not familiar enough to know about these. Would you be willing to comment on that?
It's a really good question, Rachel. And I think we have all the same problems and maybe even more, because there are things like cell lines that can be corrupted. There's some like notorious cases where cell lines were not what you thought they were. So the assay had absolutely nothing to do with the biological question you cared about. There's definitely ongoing major challenges in this space. I will say it's very hard to pull data from like different experimental setups. In many cases, I see Gabe nodding along, and he might have some real world horror stories for us there. It's often like, if you can set up one clean system that will run a collection of assays and as tightly controlled a collection of experimental settings as you can, that's really the highest quality data source. Pooling is hard. And let me punt over to Gabe, because I think he might have some good comments for us here.
Yeah, absolutely. Yeah, Tom, I was just reflecting a little bit on your comment about federated learning. I mean, I think it's a beautiful framework that machine learning has given us over the last few years, and it's made a lot of impact on, to enable privacy in machine learning. But one of the big challenges when you deal with a chemistry application, of course, is the fact that when you have even the same type of assay, but run at multiple different sites, you may have systematic differences in the readouts, which actually can cause problems for doing supervised machine learning. And so I think that's probably the biggest challenge, as Rachel is alluding to, is the fact that you cannot simply just typically you cannot simply just gather data from different assays and just pull them together and train a big supervised machine learning model on it. Yeah. It's also what makes the federation aspect intriguing but kind of tricky is that, so in my sort of day-to-day work as a data scientist at Reverie Labs, it is incredibly important to have access to the underlying molecular structures. If those are obfuscated in any way, it just makes the data science work quite tricky. It's really important to understand the chemical matter that you're working with. So I'm not saying that it would, never be able to happen. That maybe there's some clever application, but in a sort of real world drug discovery setting, it's just so critical to be able to, excuse me, to be able to look at the molecular structures in question, possibly in tandem with non-machine learning approaches. So using a molecular dynamics or physics-based simulation approach. So yeah, many challenges that are quite unique to this domain, I would say.
And I think our chair, Tom, I think you've probably similar the ball back. Well, not to put it on the spot there, but I think it's definitely a challenge, kind of real world data gathering, real world kind of like, and a part of the driver behind like efforts. Sorry, am I frozen? Can you folks? Sorry, can people hear me?
You could read.
I think it was probably me. I think I'm back. I see like some moving faces. But okay, maybe that's just a good sign from the universe that I should hand over the mic to my colleague Seon. Seon will lead the discussion of our ChemBERTa architecture and our code tutorial walkthrough. So over to you, Seon.
Cool, thanks Bharat for the great intro and overview of transfer learning. So I think given the availability of millions of these SMILE sequential data, as Bharat alluded to, I think that the transformers really offer this really interesting alternative to say an expert crafted fingerprint or a graph ComNet learned fingerprint. And so in particular, the mass language modeling task, which is really popular with these transformer architectures, we found was really analogous to an atom or bond masking task that's being used in graph settings. So to give an overview of how mass language modeling works, given an input sequence, you tokenize it into a set of essentially groups of characters which form the input vector representation. And your goal is to randomly essentially mask 15% of these input tokens and get the model to essentially recover the identity of the tokens. So by doing this, you're essentially able to make use of all the structural data that's available in these larger virtual libraries. And so while this is a really simple concept in general, I think there are several questions that are still unexplored within this area. So one question is, how does the pre-training data set size for this language modeling task impact the downstream performance on molecular property prediction? The next question that we sort of aim to answer is looking at different ways of tokenizing these molecular string representations. So like I mentioned previously, tokenization basically alludes to, given a SMILE sequence, how do we effectively split it into groups of tokens or a set of tokens? And so there's several different approaches to that. And the final question that we sort of aim to answer is a comparison of different molecular string representations. So by, say, replacing SMILEs with the selfies representation, which I believe is one of the upcoming talks in the SciML webinar, does this improve performance? And so we sort of aim to go out and answer, or try our best to answer some of these questions and evaluate the transformer architecture for molecular property prediction. So to give a brief overview of existing work in pre-training, basically for molecular representations, one of the more prominent graph-based pre-training strategies is SNAP, which Bharat briefly talked about, which is by Yuri Leskovich's group at Stanford. And when you look at one of the pre-training tasks they use, the attribute masking task, what they essentially do is, given this molecular graph, you randomly at any given time, you mask an input node or an edge, and the GNN is basically asked to predict them. And we thought that this was a really analogous pre-training task to the mass language modeling task that I talked about before. And so we sort of adopted this language modeling task from RoBERTa and applied this to a set of 10 million molecules from the PubChem virtual library. And so building off of this sort of task of atom or bond filling, we sort of hope to see if the model can learn a general understanding of chemistry. And so to give a bit brief overview into sort of the origins of this work, previously in around April of 2020, our collaborator Gabe Grant from Reverie Labs originally published this tutorial on training large-scale NLP transformers with tensor to tensor for practical drug discovery. And around the same time, we released the first ChemBERTa tutorial in Deep Chem, which basically at the time pre-trained a model on a really small subset of 100,000 molecules or smiles from the zinc set. And so building off of this, we built off this work and trained it on a data set that's several orders of magnitude larger. So in this table, we originally applied it to 4 different tasks from the molecule net set. So this is a bit of an outdated chart from our original archive paper in October. And so we applied this to BBBP, which is basically blood-brain barrier penetrability, the Clintox data set, HIV, as well as the TOX21 data set, specifically the SRP53 task. And so all of our baselines that we sort of use in this work are from the ChemProp library, which I know was discussed last week. So we use the ChemProp signature MPNN architecture, as well as the random forest and support vector machine models from ChemProp, which use handcrafted fingerprints. And so when you look at these sort of initial results, ChemBERTa does approach, but it doesn't yet beat these really strong baselines that are set by the ChemProp library, with the exception of the TOX21 P53 task. But in a later table, we'll show some improved results that show that we are getting closer. But what sort of forms the main, I think, highlight of this work isn't the fine-tuning results, but we found really compelling evidence that by scaling pre-training on progressively larger data sets and virtual libraries, we result in a really strong boost to the ROC and the PRC. So in this sort of scaling graph, we sort of look at comparing a model that's pre-trained using this language modeling task on 100,000 smile strings, and then we compare that with models that are progressively trained on 250,000 smile strings, 1 million smile strings, and then 10 million smile strings. So most of the data is from the PubChem set, which we open sourced in MoleculeNet. And so I think the main result that we see from this is when we scale this pre-training language modeling task over 2 orders of magnitude higher, we see sort of an 11% boost to ROC AUC on average and a 6% boost to PRC on average. So sort of taking a look at this graph, we sort of split it by the specific task. So orange is for the Clintox task, blue is for the TOX21 task, and then green is for the BBBP task. And then the dashed lines represent the increases or change in delta for the PRC score, and then the blue represents the change for the ROC score. And the blue highlighted region that is in the back of this graph sort of represents the mean average change to AUC as well as PRC when evaluating these models from 100,000 to 10 million smile strings. So the reason why I think this is really promising is when we continue to further scale this work, say on the entire 77 million set, or even larger virtual libraries, like thinking about the enamine real data set, which is 1.5 billion molecules. I think the main highlight of this work is that we hope to sort of explore a more broader chunk of chemical space that will hopefully continue to contribute to gains in our molecular property prediction performance. So the next part of the work aims to look at examining the impacts of different ways of tokenizing these molecular string representations, as well as a direct comparison of different ways of representing these molecules. So initially in that scaling graph that we talked about in the last slide, we used something called a byte-pair encoding strategy. So the byte-pair encoding strategy works quite well on these language model tasks because the goal is to essentially optimize for constraining the number of input tokens. So previous research in natural language processing has shown that when you lower essentially the number of input tokens, it's a known way to really boost the performance of these models on language modeling. And so this is like work done by OpenAI in their original scaling laws paper from last year. So taking a look at some of this data, we decided to apply this algorithm at first on the RoBERTa architecture for mass language modeling. And so you can kind of see here how it sort of allows you to essentially, given say a molecule like halicin from the ChemProp paper, it essentially compresses certain atoms and bonds together into single tokens. And it's completely random. It's not based off of actually the chemical significance of the token, but essentially how it does so is it essentially initializes a vocabulary, which is meant to represent essentially given patterns in the pre-training data set, you can merge it into a single token. And so you have this large vocab of say 5,000 different patterns in the PubChem 10 million set. And then using that, the model will then look at the fine tuning set and essentially merge groups of characters together into single tokens. And the main advantage of this is not just constraining the input size, but it allows you to, especially when you deal with larger synthetic molecules that are say above 128 or 256 characters, it allows you to still learn a really rich vocabulary and make sure that every single atom and bond is unlikely to be forgotten. But the main downside, I would say, or disadvantage of this approach, when you look at this example, is you actually sacrifice encoding a real atom or bond level feature, since many of them are grouped together in the process. So the second tokenization strategy we tried was essentially the SMILES tokenizer. So this SMILES tokenizer, which is open sourced in the DeepChem Featurizers API, essentially uses something called a regex, or regular expression. So the way sort of regular expressions work are you have a set of different characters which indicate when to split, which basically indicate when to split a given character or group of characters into a new token. And so by doing this, you're able to essentially directly map bonds and atoms into the tokens. And so the main downside of this approach is you do have to use 2 to 3 times as many tokens as with the BPE model. So we sort of face this trade-off between these two tokenizers where will this more chemically relevant tokenization actually replace the effects of constraining the input size? And that was the main experiment that we were looking to try. So in addition to this, we make this tokenizer open source in the DeepChem package. So it's available in the Molecular Featurizers API, and it's also used in the tutorial. So if anyone wants to try it, it's completely open source. So what we found is that SMILES tokenizer actually performs far more better than BPE on all of the tasks that we benchmarked. So even though it's using a larger number of tokens on average to encode every single SMILES sequence, the model is clearly able to learn a stronger molecular representation based on this atom and bond mapping basis. So when we contrast, essentially looking at this table, the first row essentially is the BPE model. So we first compared the 2 identical ChemBERTa models. They're both trained on 1 million SMILES strings from the PubChem virtual library, and they're both trained for the same amount of time and then fine-tuned on using exactly the same procedure. So it's basically an apples to apples comparison. And in each of the benchmarks, we found that the SMILES tokenizer beat the benchmark quite significantly across Tox21 and Clintox and BBVP. And also our model is far more competitive with the ChemProp MPNNs on Tox21 and BBVP, but we're still not there yet when it comes to beating ChemProp. But sort of our hope with this is knowing that the SMILES tokenizer is a much more better featurization strategy. We hope to sort of scale this approach and pre-train using this tokenizer on, say, up to 77 million molecules from the PubChem set. So in addition to SMILES, we also experimented with the selfies, or self-referencing embedded strings representation. So selfies is kind of this robust grammar-based molecular string, which in a bunch of generative modeling architectures for molecular inverse design, it's shown to be 100% valid. So when it's being applied to VAEs and GANs and normalizing flows and genetic algorithms, it's found that the generative outputs are 100% chemically valid, despite using a string-based architecture. So we wanted to see, looking at this representation and applying it to the BPE algorithm, could we report similar boosts seen in the generative modeling tasks? And when we compared the two, we found that essentially apples to apples, they both performed around the same. So we didn't report the same boost, strong performance boost that we saw with SMILES tokenizer. And I think one of the biggest reasons why is going back to what we found with the SMILES tokenizer. By using the BPE algorithm, we're not actually able to encode this in a way that's chemically relevant. If you look at, for example, a selfie string, like the string that's included here, each single atom-based symbol or each side branch or aromatic ring is signified with brackets. So if we could develop a new tokenizer that essentially makes use of this and tokenizes every single atom or every single essentially atom or bond as it traverses through this molecular graph, that might be one way to actually report those same boosts. And so this final piece of work that we've been working on is designing a new pre-training task, which uses basically the SysChem descriptors as a prediction task. So this is essentially a pre-training task that's been used by several architectures in the past. So papers like Chemception make use of RDKit descriptors, as well as a similar paper called MolBERT, which applies this pre-training task to virtual screening. And so to give a brief overview, essentially we computed 200 different RDKit physicochemical descriptors for all 77 million PubChem smiles. So some of the features that are learned here include the molecular weight, the QED, the number of rotatable bonds, and many more. And so essentially you can think of this as a multitask regression, basically predictive task, where given a smile string, we are asking it to basically predict all 200 of these different descriptors. And I think the most promising the reason why we wanted to try this approach out was one of the big downsides of the mass language modeling approach is sample efficiency. So for every single smile sequence, you're only able to mask 15% of the tokens at one time. But when you try at the same time, if you try something like this pre-training task, for every single given smile sequence, you can run over 200 different predictive tasks, which hopefully will allow the model to reinforce a better prior molecular representation before then applying it to molecular property prediction, and also hopefully do it more efficiently using less smile strings. So as a final part of our work, the last part of our work sort of goes into interpretability and trying to understand how the attention mechanism is applied to different molecular structures. So originally, we tried looking at how attention was applied to graph attention networks, where essentially the goal was to see, was it prioritizing certain substructures, for example, in the TOX21 data set, that were indicated as being important towards making a prediction for binary classification on the TOX21 data set. So this could include highlighting a substructure like a ketone or a functional group, or for example, an aromatic ring. And so applying this principle to the SMILES tokenized inputs, we essentially generated this 2D demo that basically looks at how this model is performing, and for every single given input token, what neighboring tokens is it paying the most attention to. So we used a library called BERTviz to sort of inspect all of the attention heads. So in ChemBERTa, we have 12 different attention heads spread out across the 6 different layers. And so we found that certain neurons were actually selective for chemically relevant functional groups like ketones, and as well as aromatic rings. And we also observed other neurons which actually tracked a lot of the relevant SMILES grammar. So things like bracket closures and openings. And sort of to give an idea of how this diagram on the right works, essentially given the query vector for the input token on the left and a key vector generated for the token on the right, it's really just computing the dot product of the two and then applying a softmax normalization to sort of compute this importance score. So the goal of an attention model here, that to really show that your model is learning something, is you want to make sure for every single input token, it's not just highlighting all of the neighboring tokens with a uniform distribution. It's actually paying attention. You want it to essentially pay attention to specific tokens, such as a bracket opening or bracket closure or neighboring tokens. And so that was sort of the importance of this work and something that we were looking for. So the next part sort of goes into open sourcing the language model. So within DeepChem, and using this library called Hugging Face, which is this really excellent natural language processing library, we were able to open source all 15 of the ChemBERTa models, which essentially is what allows us to use our tutorial and allows anyone to essentially load the models directly from the API. And so far, the models have been used over 200,000 times through the Hugging Face API. And so I think following suit with one of the goals of DeepChem, which is essentially trying to give more access to some of these scientific tools for molecular machine learning, we try to open source all of the models as well as the tokenizers and the pre-training procedure. So now I'm going to briefly go through the DeepChem code tutorial. So essentially, this tutorial is completely open source in DeepChem. And in this tutorial, what we're going to be going through is using ChemBERTa models to essentially apply towards toxicity prediction on the Clintox data set. So it's completely runnable. So it's within Google Colab. So within the DeepChem tutorial, you can basically open it up in Google Colab and generate your own runtime instance and go through the tutorial. And so we already pre-ran it mainly because some of the imports take some time. But essentially through the tutorial, we essentially aim to do 3 main things. So the first thing is looking into mass token inference, or basically running a sample language modeling task on a file string. the next thing we aim to do is sort of examine some of the attention heads of the ChemBERTa model. And the final thing that we do in this tutorial is essentially fine-tuning our ChemBERTa model and the SMILES tokenizer models on the Clintox toxicity data set. So the first part of this tutorial I kind of want to highlight is the mass language modeling. So given, for example, a really simple SMILES string here, we wanted to predict this specific character. And so I think what's interesting is it is able to reconstruct and identify the correct SMILES character, but all of the following predictions that it makes with a lower confidence score are actually all chemically valid. And I think that this is important because it's not just a model that is essentially inputting any random ASCII character as a prediction, but it is actually generating permutations of this SMILES strings, which are also chemically valid, which we checked in RDKit. So the next part of the tutorial sort of walks through attention. So we wanted to approach this from the perspective of someone who is both new to NLP and molecular machine learning. So we try to go over some of the main concepts of attention by sort of using the analogy of the attention mechanism being something like visual attention, where, for example, when you're reading a book, your focal vision is paying attention to that book, and it tends to not zoom in as much on your surroundings because you are paying attention to that book. So you can kind of think of that analogy towards a SMILES string, where you wanted to essentially highlight the most chemically relevant substructures that make up the specific prediction that you're trying to do. So here we run a demo of basically the attention visualization. So within here, you can basically peer through all 6 of the different layers and try it on a sample SMILES string. So the main thing, like I mentioned before, that you want to see is that for every single input token, the neighboring tokens that it's paying attention to are not just uniform across all of the essentially neighboring tokens, because if it's paying an equal distribution of attention to all of the neighboring tokens, then there is no point in using attention, because the main motivation of an attention-based model is to make sure that it can sort of overcome like an information bottleneck and prioritize the most relevant substructures in making a prediction or forming an embedding. So in this tutorial, you can basically try any single SMILES string and run through this demo. So here, this sort of figure is sort of our different general model views. So for every single one of the 12 attention heads and 6 layers, we computed essentially a different attention diagram. And you can kind of see here that while in some specific diagrams, it's paying more uniform attention, but in some it pays very targeted attention to specific tokens in the SMILES string. So I think this sort of reinforces that concept. So the final part of sort of this code tutorial is fine-tuning ChemBERTa on the Clintox dataset, which is, Clintox is essentially this toxicity data set. It's around 1,500 binary labels, and it's basically a set of molecules that were in FDA clinical toxicity trials, and it's just a really simple binary classification data set. So here we are using essentially a MolNet data loader, which our colleague Gabe built. And so it basically allows you, similar to how the ChemProp library allows you to load a CSV and then run a prediction on that. Essentially, it loads the CSV version of the molecule net, of any molecule net data set, and then applies scaffold splitting. So you can do like an automated fine tuning. So using this tutorial, the one thing that we also wanted to reinforce is you can plug and play this with any single one of the molecule net data sets and any one of the 15 models. So there are hundreds of different permutations of models and data sets that you could try to fine tune on. So first off, we benchmarked this against the original BPE model that is pre-trained on 10 million SMILES. And so we were able to essentially, once again, use the Hugging Face API to load these models in automatically, and then fine tune that on, and then we fine tune this on the Clintox data set. And what we find is we achieve around a 97% ROC PRC for Clintox. But then when we try the SMILES tokenizer, which is also loaded here later on in the tutorial, and then fine tune it, what we found is that we achieve around a 99% or 98.9% ROC PRC. So this basically was sort of a written example. And I think the main goal of the tutorial is just trying to show how to interact with some of the different modules within the ChemBERTa infrastructure, to how to fine tune models, how to load models from the API. And overall, it's just sort of a really introduction to how to sort of play around with any of the different features in the ChemBERTa paper and generate the figures for yourself. So yeah, that's, I think, the code review.
Super clear. Thank you so much, Seon. We already have one question from the audience from Kanpu, who thanks you for the great talk, and points out that you used canonical smiles when you called PubChem. And do you have a strategy for distinguishing isomers?
Mm-hmm. That's a good question. That's actually a pretty good question. I'm not sure if we've actually tackled that. Maybe.
Well, so it depends on the data set, right? If the stereochemistry information is present in the smiles, then we preserve it. It doesn't get destroyed in the canonicalization process. If it wasn't there, then we don't have information. Barth, I don't know if like all of the molecule net subtasks have been checked for whether they have the stereochemistry information, but I'm assuming that most of them would.
That is an excellent question. I don't know offhand. I think some of them would at least, but
Yeah.
not sure offhand.
Yeah.
But this is a very important question from Ken because the stereochemistry does matter a ton, and it can have significant changes in functional activity.
Absolutely. Okay.
Great. I'll ask a question to you, Bharat, and perhaps also to you, Seon. So I observed that as you jump towards selfies and smiles, there's also a jump in performance. And perhaps we're saturating between selfies and smiles. So if you were to move towards a more expressive representation, say beyond selfies or smiles, what are perhaps the most important aspects that you want to incorporate? Is it, say, bond distances, torsional angles, magnetic moments, or what would these be from all the experience so far?
That's a really good question. And if I had to pick one thing offhand, I'd say actually Grover, which is a paper from NeurIPS 2020, would be the direction to go. So Grover essentially does, I'm doing this from memory. I think it's like the RD-Kit style pre-training task, but applied to graph networks as a base. I believe right now Grover has actually achieved state-of-the-art and is even better than ChemBERTa. Sorry, not ChemBERTa, I mean ChemProp across the board on a variety of different tasks. Now, the only downside to Grover is that it's very, very compute intensive. This is from a Chinese group, I believe, at Tencent. And in big tech style, they, of course, ran it on a bajillion GPUs, which I think our hope would be, is there a way to see the Grover-like performance boost while maintaining the simplicity and efficiency of the transformer architecture? But yeah, I think our guess is that we want to move ChemBERTa closer to, as you can see, like we started a good ways below ChemProp. We're still not quite as good as ChemProp, but we're kind of within the kind of neighborhood now. Grover, I'd say, is a little bit higher up yet. So the question for us is that how do we close that gap without, say, losing the simplicity, ease of use, and efficiency of the ChemBERTa style architecture? But still an open problem, I think. And we'll also punt over to Gabe and Seon, who might have I want to add on there.
Yeah, so I think with the graph transformer, it's a really interesting idea, which we were also exploring, mainly because you're able to inherently use the molecular graph representation. So you get over, it sort of is natively representing a 2D molecular structure, but you also sort of are able to embed attention layers. The main downside is obviously it is compute intensive. They use 200 GPUs and they train for 3 weeks on 200 GPUs. So it's, I think, exponentially longer pre-training time than our model. So you can see, I think it's pretty evident that seems to be a really strong representation, but you sort of, all sample efficiency sort of goes away when you go with those models. And I think answering your question about selfies, I think that's also a really good question, because originally the motivation for using selfies was it embeds a lot of language, like for example, like there's a branch or ring or all of these sort of keywords that you think that the transformer can hopefully pick up on when it is sort of pre-training on this corpora. And I think sort of looking at our results, we're achieving around the similar results as BPE, but they're not exactly seeing the same boost that we saw with the SMILES tokenizer variant. And I think that sort of goes back to answering how important how you tokenize your input representation and how your featureization strategy really from an NLP perspective is, it's in our case even more important than, for example, training it on 10 times as many molecules. And we're like, with that PubChem 1M model that's trained on 1 million molecules, we were able to beat the ChemBERTa model that was trained on 10 million molecules using the BP tokenizer. So I think there are a lot of interesting ways that are sort of very underexplored, actually, which sort of go into the representation and the novelty of the pre-training task. And I think, in my opinion, compute is definitely the one answer which OpenAI and big tech and all of it's like the most evident answer. But I think there are a lot of ways for people to get around those improvements by sort of innovating on the really low level, simple functional stuff. So that's sort of my opinion.
I think now's a good time to jump in and give a plug for the March 4th contribution to this series, which will be coming from Mario Kren of the Sprugusic Lab, who will be focusing on selfies as well. So for those who are interested in learning more about that. I think a lot of people are curious about just coming to this talk and learning about transfer learning, just having a sense of the things they should be worrying about as categories about whether or not transfer learning will work in a given case. And, if one tries to break that down, you might say there's the actual task with respect to which you're transfer learning. There's the representation and model you're using for that transfer learning. There's the data set that you have available to you. And which of these do you think is the most limiting in terms of, and are there additional dimensions that one needs to be worrying about in terms of those three categories?
That's it's an excellent question. I think short answer, it's really hard. In the past, so one project I did, so in an earlier iteration of like DeepChem's transfer learning efforts, we had a wonderful Google Summer of Code student, Vignesh, who's now doing his PhD at ETH Zurich. He explored the use of RDKit descriptor style pre-training applied to these what are called conception architectures. You treat the molecule, you turn it into an image. It's a beautiful it's a beautiful hack, I think, is how I would describe these architectures. I really like them. Like you just turn it into a picture. You're like, I know how to do image learning. I know molecules. I can turn a molecule into a picture, like in a chemistry textbook, bam. And it actually ended up working quite nicely when we benchmarked on a few different data sets from MoleculeNet. But then I went out and kind of tried it in a real world project with a friend who was at a biotech, and we were able to replicate all the results from the paper. That was totally fine using Chem. But then we tried it out on their in-house data, and it just like nothing worked. And we couldn't really understand why. So one of the big questions that we want to continue iterating is that we've set up some infrastructure to begin testing ChemBERTa across the board on all of MoleculeNet. So I think I don't really have an answer in terms of like what would make it more robust, able to handle more categories. The best I have is an empirical answer in that by setting up the infrastructure so that we can test the hypothesis of, is this at least pan-molecule net transferable? I think that will at least enable us to do design search over, architecture, models, representations, featurizations that can make a difference. And I think like the Smiles tokenizer work in one sense is, an example of our architecture playing early dividends. Because we've begun to set up this more systematic infrastructure, we are able to benchmark at larger scale, but we have a long ways to go. I will also do a brief plug for the MoleculeNet V2 effort. So we now have, actually, Seon, would it be okay if I grab the screen for just a bit? I might see if I can just share. So while I'm kind of pulling up pulling that up. So we are running the new MoleculeNet benchmark suite as a community-run benchmark. So we've got folks from kind of multiple groups contributing in essentially benchmark runs to the new MoleculeNet. This is something that we aim to get ChemBERTa in. Let me see if I can share my screen here. Anyways, this is unfortunately defeating me on the text, so I will just drop this in the chat and you can all check the link out. So we have like a community kind of run leaderboard. We have researchers from multiple groups, a couple people from AWS, something we aim to add on ChemBERTa as well. I've been talking to some of the other leaders in the field to get them on board as well. But I think basically coming back to your question, Tom, broad testing across a wide range of chemical tasks seems to be the only way to like test the robustness of the transfer learning hypothesis. And even then, I'm sure the minute we put this out, someone will be like, hey, this is great, but I tried it on my one weird assay and it all blew up. And it's the unfortunate molecules are infinitely malleable. Well, not infinitely, but extremely malleable. And it's just hard.
Yep, that's very thoughtful. I definitely want to encourage other people to chime in with questions. I'll nudge that one as well. Just that other question. You know, part of the strategy for generating these big pre-training data sets is enumeration, as you point out, right? The enamine real and so forth. And those are, you know, that enumeration uses a particular logic for the enumeration. That particular logic expresses a particular representation, which might in some sense be redundant with the actual representation that is eventually used on the machine learning. So I wonder if some element of the saturation that people see from the performance of the pre-training is due to that redundancy in the representation you're using with the way in which you're actually creating the pre-training in the model or in the data set.
So Tom, we've been really interested in that at my company at Reverie. For this particular work, we were able to put together a data set from PubChem. So PubChem, in contrast to something like enamine real, where you might have, you know, billions of molecules, but they've all been created from the same set of limited building blocks and same set of combinatorial chemistry. PubChem is, I think, a much more naturalistic and representative, sample of chemical space. And so the hope is that by doing pre-training on that it'll lead to better results. But that is definitely an interesting consideration that the chemical, you know, even you might have a lot of unique structures, the actual chemical diversity might be limited because it is being created across specific avenues.
Very good. Any other questions?
Tom, perhaps this is a good point to ask you for your industry perspective, since you're more in the real world. So, you know,
goodness help us. Goodness help us if my 10 months of industry perspective is what we have to draw on, but I'll take a stab.
I should make a plug here for Tom. One of the VCs that funded Tom, he said that we really got this one. It's amazing that we got to fund Tom's round. Extraordinarily excited to hear your take on how these 10 months have been. It's been it's always incredible to see tenured faculty member taking, I guess, your sabbatical wisely or maybe not, but would really love that perspective, Tom.
Oh, sure. I think that we're all after the same things. We want to see our science impact real world problems. And I think this field is really at the cusp of that in a way that's tremendously exciting. I think, we all develop our algorithms, we develop our methods, we write our papers, but we want to see that converted into things that make the world around us better and things that actually translate to real technologies. And I think entrepreneurialism is a great way to do that, and doing that in a way that is compatible and leverages and mutually beneficial with the academic community is a tremendously important thing. And we've seen those themes throughout this talk. We've seen and we see that every day in our own work. So I think it's an increasingly permeable interface. I think academics and the startup world. It's been so exciting to have a chapter in my own life where I've been able to try that out, and it's been great fun. And I'm excited to continue that. But I think everybody here has had illustrious academic careers and is dabbling in different ways in that other world as well. So I think that we'll just see more and more of that. And this is a wonderful place for that transition to be taken with rapidity and effect. Bit of a hallmark card response, I'm afraid, but I can ask if you want to ask a more pointed question. I can answer that too. But you set me up with a softball, and I give you a softball later.
No, I think actually, no, this is great, and I think one of the visions for this series even is sort of create that full ecosystem, right, where we have obviously amazing speakers. And also try to have ideas across domains, and we also have a VC panel coming up in a couple of weeks, so we'll have people that hopefully can cut some checks that also see the people that are here, and I think we're building a pretty incredible community here. I know Dilip has sort of helped curate a great series here, so I think, as you pointed out, Tom, I think all of us are sort of playing in this space in various ways, and I think the challenges that we have need to be solved at a much more accelerated timescale than the Edisonian approaches we've been going after. So it's quite exciting, and I'm glad that we're able to sort of curate this package here, and hopefully this will see new companies and other things spinning off from here.
Yeah, absolutely.
I think to add maybe like one bit of color here, I think that part of the reason for the shift towards, say, more industrial settings is just the compute data requirements. With ChemBERTa, we're still really in the shallow end of the pool for how big these things can get. Like if you look at the extraordinary infrastructure put together by OpenAI, by kind of like Google for some of their systems, it just takes a lot of effort to build these things out. So we, I think we've had to be very creative with kind of cobbling together resources. I think at one point, Seon stitched together multiple Google Colab runs, and we had a beautiful hack to get that all kind of tied in together. And I think the reality is that, if you look at a team like Grover, at Tencent, of course, a little bit outside the usual American ecosystem, big tech, they just have access to, say, way more resources. And it is a challenge, I think, it is quite possible to do very meaningful work, I think, in this space without access to those big resources, but it does kind of force this, extra barrier of creativity needed to get off the ground, which is both good and bad, but I can see why it might be possible. And we've seen this with like Facebook AI, Salesforce AI, and Google have been putting out these really great protein transformer papers. They basically downloaded Uniprot, stuck it on many GPUs, did many interesting things. It's really good science, but it's also a place where, their labs have a unique structural advantage that would be quite hard for most academic labs to compete with. But I think part of our goal with DeepChem is to enable enable these types of experiments to become more accessible. But we will say it's a hard challenge. And I think there are genuinely many scientific questions that being in industry, you might be better positioned to answer, or maybe not even the small industry, big industry like big tech. Or if you have, say, from Venkat's VC panel, enough checks, maybe that also could solve that problem.
I think that's a really interesting point. The challenge of, just the infrastructure that a big company has versus an academic lab does enable you to do different things. And that is a story, of course, that is a very old one. Dow Chemicals always had a lot more chemists than any individual organic chemistry lab, right? And yet that has never hindered universities and academic groups, despite the fact they have smaller groups and not only less resources in that way, to drive innovation. And there's nothing unusual here or different about that here, except for the fact that, something about computation and software is more easily scaled than than actual hardware and hoods and all that sort of thing. So, you know, we might have an opportunity here, and I think you guys are doing wonderful leadership in that way to use the unique aspects of software and computation to reduce that intrinsic gap that is existing in so many fields.
And I'll do kind of a brief plug for like DeepChem in that. So part of our goal is to create what we're calling like basically a decentralized research community. I don't, wait, no, Gabe and I met in person once, I believe, but Seon, I don't believe you've ever met either me or Gabe. Like just having kind of an online, purely online, like, you know, nonprofit, like, no, it's an open source community. So as with open source communities, it's not even quite a legal entity. It's just a thing on the internet. Has created, I think, you know, I've been excited to see the emergence of like great research like ChemBERTa, just from like drawing in smart people and getting them to talk to each other. So my hope is that like as like more of the world moves more online, like Venkat's and Dilip's like excellent seminar series are just drawing us all here, whereas previously might have been limited to the benefit of the CMU community, but now we have everyone from all over kind of joining in. I think that there are these like really interesting like new opportunities that for like very interesting cross-pollinations to emerge that we're beginning to see from this new way. So this is something we try to actively encourage at DeepChem, getting researchers from like, so we have like students from Tokyo, we have like, you know, researchers in Zurich, we have high school students in Canada, to say on a shout out, like, and who are all able to do science together. So that I think is one of the really nice bits of, say, this type of open infrastructure. And I think, yeah, going back to your point, Tom, I think you're absolutely right. This is a very old problem. Large industrial settings have always had the ability to do experiments and you know, answer questions that small groups have not. And as you rightly point out, that hasn't stopped creative scientists from like, you know, making lemonade from the lemons of small resources and doing wonderful science.
Yeah, or taking advantage of the freedoms that don't come with that infrastructure as well. Yeah.
Yes.
Yeah.
Can I jump in with one more in this, and hopefully we can move to more questions. But how do we sort of commercialize here, right? You know, ultimately, you know, someone has, I mean, and this is something Bharat, you made a comment in one of the earlier webinars that the reason DeepChem has been extraordinary, you know, has been where it is, you know, you have not had to rely on a dollar being earned. You know, you are not dependent on any revenue from DeepChem, right? So, but how do we sort of, you know, what's the right way to think about the commercialization model with so many models, code, science out in the open, right? You know, how do we sort of differentiate, you know, as a business? And maybe, you know, maybe Bharat, you can take it, and maybe even Tom, if you can share what you've sort of seen in the last, you know, 10 months or so at Entos.
That's a great question, Venkat. I think I'll give kind of two answers to that. I think the first answer is that there's this, I'd say, really an argument that open source should be better supported by grant writing, creating agencies. There's just not enough grants that actually recognize like open source software. There's some standouts that this has been beginning to change. I think Chan Zuckerberg Foundation in particular, I think, has been starting to fund open source software, which is very sure from their parts. So I hope that you know, more traditional federal funding agencies in the US and elsewhere begin to recognize that supporting open source software is a very good use of kind of grant resources. Coming back to the second point, I think with DeepChem we've like tried to maintain like a neutral, common, shared commons essentially. So there's multiple companies that use DeepChem for various things. Gabe, of course, is working at Reverie Labs. I have my new company, Deep Forest Sciences. I know there's a couple other startups that use Chem for various purposes. And I think that at least for DeepChem, I think the neutrality serves the purpose of kind of creating freedom for each group to kind of like use it in a way that makes sense for their purposes. I know there are a number of startups that have at least gotten their start with deep chem to bootstrap off the ground with their initial models. So I think we've helped a lot of different people get off the ground. The flip side, there isn't saying like, because there isn't one commercialization strategy, it does get harder to scale. Like if say like we co-founded a deep chem company at the very beginning alongside deep chem the open source, it might well have attracted more resources that say Hugging Face style could have enabled us to get this to more people. So I think it's a collection of trade-offs. I can't say that this is like necessarily better or worse than another collection, but I think that the way I think about it is we fill kind of a unique niche that no one else has quite occupied. And I think sometimes that's valuable in its own right by just offering another option on the table for other folks. But then let me here punt over to Tom, is I'd actually be really curious to get your take, Tom.
Yeah, I mean, when one asks how to commercialize, there's as many commercialization strategies as there are business models and as there are actual products. And I think one thing that comes down to it is what is the actual thing that you're seeking to commercialize? What is the real point of value? You know, as Bart pointed out almost on slide 2, drug discovery is a very long pipeline. And the value generated at different phases is very differential. So, I think there's a lot of leverage to identify parts that need to be kept internal, and then parts that would be more beneficial to the community to be as open as possible, so that you can contribute to that and in turn benefit from that. In our own work, we already find that even on the short time scale, there are some things that we don't open source, but there are some things that we've fully embraced in terms of not only provision freely to the academic community, but also open sourcing as well. You mentioned the Chan Zuckerberg, the Eric Schmidt Foundation, for example, has given us some support where it's expressly for open source endeavors too. So these things are not incompatible. I think each company has to have its own business model. Each commercial enterprise has to have its own things that it holds dear and then shares with the community. And striking that balance in a way that's the best for the broader community is something that the right answer is even time dependent, right? A company at a certain stage might make one choice, and then at a later stage might be able to shift what is more natural to keep in house and versus external. And I think we'll always see that.
I think to raise an amusing point there, Microsoft, the villain of Linux in the early days, is now what supports GitHub Actions and all these other tools. So yesterday's villain can be tomorrow's hero, and vice versa, a little bit more darkly. But yeah, I think I think open source nowadays is big business. I think that there will likely be patent issues that arise at some point in the future from all these efforts. But I think it's also part of the messiness of science is that it's unavoidable. Like, I think being afraid of like dealing with the complexities of these issues like could well prevent us from doing meaningful work. So I think we have to I think you said it really well, Tom. I think we have to try the best we can, like reach a balance and see what makes sense for each kind of company, each organization.
Wonderful. Well, we are the panel has been sucking a lot of oxygen out of the air. If there's any time for and I acknowledge my role in that. If there is any remaining Q&A that people want to submit, I'm sure that would be enthusiastically received. But in the meantime, as we wait for maybe that last question, if it's going to come in, Dilip wanted to make an announcement before we wrap up.
Excellent. Okay, so I'll just make a quick announcement for the following 2 weeks. So the next couple of weeks will be the last 2 talks of the series on molecular machine learning for drug discovery. So Feb 25th, next Thursday, we have Dominic Lem from Anatole von Lilienfeld's group from the University of Vienna. And the following week, March 4th, we have Mario Kren from Alana Spuru-Guzik's group, University of Toronto. So with that, I want to thank Tom for taking the time out to chair the session. Thanks a lot, Bharat, Sayon, and Gabe for such an amazing session and the interesting discussions. Everyone, please stay tuned and thanks for joining in. See you all next week.
Wonderful. Thank you, Dilip. Thank you, Venkat. Thank you, Bart. Thank you, Sam and Gabe. Bye-bye.
Thanks a lot, everyone. Bye.
Thanks so much.
Thanks a lot.